Welcome to Nervous System Part 2. I'm Debbie Richards, and today we'll explore how a neuron works. I hope that you have with you your student note page as well as a pen or a pencil to write with. And just as a reminder, you can stop this video at any point to replay any items that you think you need to review. So let's get started. Don't be nervous. Get it? We'll start by reviewing our essential knowledge statement. Animals have nervous systems that detect external and internal signals, transmit and integrate information, and produce responses. In Part 1, we looked at the general pathway of information flow, which starts with sensory reception, and then that sensory information is inputted into the central nervous system, where it is integrated, and then we have some sort of motor output. The motor neuron delivers information to the effector, which has some sort of response that it generates. In Part 1, we also looked at the structural adaptations of a neuron to see how it is different from a generalized animal cell. So I'd like for you to stop the video for a second, number from 1 through 5 on your paper, and see if you can list the structure name as well as its function for this typical neuron. I hope that for number 1, you put a dendrite. The dendrite receives incoming information. For number 2 is the nucleus of the neuron, and that's where the genetic information is stored. Number 3 is the axon, and today most of the information we'll be covering deals with events that are occurring there in the axon. Number 4 is the axon hillock, and that is where we'll have the impulse generated and the information flow starts down the axon. Number 5 is the cell body, which has the general organelles and components needed to keep that cell alive. I'm going to show you a short video that is going to cover 2 kinds of potentials or conditions that exist in a neuron. The first is the resting potential, and then you'll also hear about the action potential. And those are 2 different environmental conditions that allow a neuron to do its job well. So I want you to listen for what is the resting potential, what membrane structures are involved, and also what ions are involved. And remember that you could repeat this video as many times as you feel like you need to.
This baseball player springs into action, his nervous system rapidly processing information in a network of nerve cells or neurons. Each neuron receives input from one or more cells. In response, the neuron may generate an electrical signal known as an action potential that travels down the length of the axon. Let's take a closer look at the plasma membrane of an axon. Even without an action potential, the axon is a busy place with many ions moving across its membrane. Much of this ion movement is driven by the sodium-potassium pump. Using energy from ATP, sodium-potassium pumps actively transport sodium ions out of the cell and potassium ions in, creating an uneven distribution of charge across the membrane. Some potassium channels are open all the time, allowing potassium ions to leave the cell. As a result of these ion movements, the inside of the cell is negative relative to the outside. This condition is called the resting potential. The membrane of an axon is also packed with gated ion channels that open and close during an action potential. At resting potential, the gated channels are closed. If a stimulus changes the distribution of charge across the membrane sufficiently, the gated sodium channels open. Movement of sodium ions across the membrane makes the inside of the cell more positive. This reversal of the charge distribution causes the gated sodium channels to close and the gated potassium channels to open. As potassium ions move out of the cell, the original charge difference is re-established across the membrane, closing the gated potassium channels. This sequence of events is called an action potential. The sodium-potassium pump restores the distribution of ions back to their previous levels at resting potential. Let's look at how the action potential is conducted along the axon. As the change in charge difference across the membrane spreads from open sodium channels, other sodium channels farther along the axon begin to open. The original sodium channels close, and adjacent potassium channels open. As potassium ions move out of the cell, the original charge difference across the membrane is restored, and then the potassium channels close. Meanwhile, new sodium channels open, followed by the opening of new potassium channels and the closing of sodium channels. In this manner, the action potential is propagated along the axon of the neuron, eventually reaching another cell. The information carried by this action potential will be processed with other information, permitting this baseball player to make a spectacular catch.
All right, that's a lot of information to take in, so let's break it down into some steps or smaller components. Let's begin by describing a resting potential. What is the charge inside the neuron when it's at rest? During resting potential, the charge inside is going to be a negative 70 millivolts, and the outside would be much higher than that. And so we have this polar arrangement where there's a negative charge on the inside and a positive charge on the outside. When we come to question 2 here, why is this cell negative inside and positive outside? We need to look at some of the components that are actually inside and outside the cell. In a mammalian neuron, we have potassium, sodium chloride, and then we have some fairly large proteins. Inside the cell, the intracellular concentration of potassium is about 140 millimolar, and sodium is about 15, chloride ions are about 10, and then we have a large concentration of proteins. That gives us an overall negative charge inside the cell axon, and then outside we don't have those same proteins, so that the charge on the outside remains fairly positive. Look at the concentration of sodium inside and outside the cell. Externally, then, we're going to have more positive ions, so we have this charge difference from one side of the cell to the other, and so the cell is polarized when it is in its resting condition or when it is at rest. And then when we look at how the charge changes, let me just start the video again and show you. I'll stop at a certain point to point out a specific
This
effect.
baseball player springs into action, his nervous system rapidly processing information in a network of nerve cells or neurons. Each neuron receives input from one or more cells. In response, the neuron may generate an electrical signal known as an action potential that travels down the length of the axon. Let's take a closer look at the plasma membrane of an axon. Even without an action potential, the axon is a busy place.
All right, I want you to see that these little bumps that you see, the yellow, those are the tops of the phospholipids. And so we are outside the cell. Remember on our neuron diagram, we are outside the axon there, and then we're going to see inside the axon here as this progresses.
With many ions moving across its membrane.
There, you see the lipid bilayer. You can see the external side and the internal side. And then inside this axon is where we're going to have the negative charge and the positive charge on the outside. We see these embedded integral proteins who are going to play a role in keeping this charge gradient in place.
Sodium-potassium pumps actively transport sodium ions out of the cell and potassium ions in, creating an uneven distribution of charge across the membrane. Some potassium channels are open all the time, allowing potassium ions to leave the cell. As a result of these ion movements, the inside of the cell is negative relative to the outside. This condition is called the resting potential.
We have just seen the internal conditions of a neuron that's not firing. It is not sending a signal, and it has an internal charge of about negative 70 millivolts. So it is exhibiting a resting potential. So let's consider what changes occur to cause this neuron to go into action or to send an impulse by reaching the action potential. All right. So when we look at the action potential, that is going to be the transmission of information further down the cell. This is going to propagate the impulse down the neuron. So this polarized condition becomes really important. So the gated channels are going to open and cause the membrane to depolarize. When those channels open, sodium rushes in, and then the sodium-potassium pump will reestablish that condition. So if you would find your graph or take a moment to draw this graph, we're going to look at the different parts or events that we just saw in the video and try to graph them to show sort of a logical sequence that they go through. So we'll start with stage 1, which is the resting potential. That's when we have the voltage gated sodium channels. They're going to be closed. The potassium channels are for the most part closed, and so the internal concentration is negative. We call that condition the resting potential, and notice it's way below negative 50 millivolts, so it's very negative, around a negative 70. And then the threshold potential occurs when those sodium channels first open. We get a little bit of sodium flowing in, and that's going to cause a change to occur inside the axon. You see here at part 2, we're going to see that as sodium comes in, being positive, it makes the internal condition less negative, so the charge is moving upwards. And once the threshold potential is reached, when enough sodium has come in to reach the threshold, which is usually around a negative 50, 55, 60, somewhere around in there, when we get enough sodium to reach that charge, then an action potential will be generated. It's called an all or none response because once you reach the threshold potential, the information is going to go down the axon. It's not going to be stopped. And so it's an all or none response. And so when those voltage gated channels open and let some sodium in, during the rising phase, we're going to see that the charge inside the neuron, inside its axon, is going to go up. It's becoming less negative. And that stage between 2 and 1, here is the rising phase of the action potential. So we have a depolarization and then the rising of the action potential. The action potential causes this impulse then to be generated. We enter into the falling phase of the action potential as the sodium-potassium pumps the sodium back out into the environment outside the cell and puts the cell back at its resting potential. In fact, it overreacts just a little bit and will hyperpolarize the cell or go to an undershoot stage where there is actually the potential is slightly lower than it was even to begin with. So it drops below a negative 70. And this prevents any additional refiring or resending of the same action potential. So a cell that's not firing, not sending information is in the resting potential. And then as stimulus is received, sodium is going to be allowed in. And if enough sodium comes in to hit the threshold, then we'll have the propagation of the impulse, because this situation is a positive feedback situation. When sodium comes in, it causes more sodium to come in downstream, which causes more sodium to come in downstream. So we see this propagation of the impulse as a result of positive feedback. And so that then, when the impulse has passed that specific spot on the neuron, the sodium-potassium pumps will pump out the sodium, return the potassium to the inside. Remember, the protein stayed in place, so that negative charge can be reestablished, and they will actually overshoot, pumping out a little more sodium, and this is to prevent any refiring of this neuron, and then it's going to return to its resting potential and be ready for its next firing event. So when we look at your diagram, I want you to number the parts on your graph, and then take a moment to describe what is happening at area 1. This is the resting potential, negative 70 millivolts inside the cell. Area 2 is where we see this influx of sodium as the impulse arises. Then area 3, we have that rising portion of the pathway where lots of sodium is coming in under the positive feedback mechanism. The sodium channels are open, allowing sodium to come in. And then once the depolarization has occurred, repolarization will happen as the sodium-potassium pumps move sodium back out of the cell and potassium back in. And then there's the hyperpolarization that occurs at 5, where we have sort of an overshoot of the, or an overreaction, I guess, to the pumping out, and you actually undershoot the level or the charge inside the cell. It goes below even the resting potential. And then through some simple diffusion that's going to occur, the cell returns to its native resting potential, waiting for a refire. So you should be able to write a description of the events that allow the firing of a neuron to occur. And so using those numbered steps or events, 1, 2, 3, 4, 5, that will help you to be able to write a good, decent description. So let's look at these events and give them some names. Let's start with the refractory period. The refractory period is going to be occurring after the action potential. A second action potential can't be started then because the neuron's sort of resetting itself, and so it is going to not allow the sodium channels to work at that time to prevent any refiring. And so the action potential is going to usually be generated there at the hillock, and then that depolarization is going to occur down the length of the axon. It only goes in one direction. There's no upstream backflow. It only happens in one direction, and you can see why, because sodium comes in and then the downstream sodium channels open as a result of that. And so the inactive sodium channels behind the depolarization are going to prevent any backward flow of this potential. So if we looked at this diagram, the red line shows us this is the plasma membrane of the axon. In this area is external, the cytosol is internal, and then external on this side. So we see an action potential coming and it's generating this charge change, and it just moves down the axon, and then once the action potential has passed a specific spot on the axon, then the sodium-potassium pump reestablishes the resting potential. So see if you could sequence these events. We've talked some about all 4 of these: the depolarization, the resting state or resting potential, the repolarization, and hyperpolarization. What do you think would be the order of occurrence of those 4 events in a typical neuron? Just put your sequence on your paper somewhere. Hopefully this is what you put. That a cell would be resting, and then sodium would enter, causing a depolarization of this polarized condition. Then there would be hyperpolarization, where the charge is even more negative than it was to begin with. And then a repolarization as natural diffusion returns the cell to its resting state. So you should be able to describe these. What would you say is this area's primary condition? What's happening here? The value of the charge is a negative 70 in this region, where we would have the polar condition. Then in 2, we have the sodium influx. 3, we have depolarization. 4, repolarization, 5 is hyperpolarization, and then the cell returns to its resting state. Let's look at a multiple choice question. A and blank in sodium permeability and or a blank in potassium permeability across a neuron's plasma membrane could shift the membrane potential from a negative 70 millivolts to negative 80. What's going to cause the internal condition to become more negative? Would it be an increase in sodium permeability and an increase in potassium? Or would it be a decrease? I hope that you put C, the decrease and increase. The decrease in sodium permeability and an increase in potassium permeability would shift the membrane potential from a negative 70 to 80 millivolts. So you have to let your brain think about that for a minute. Sodium's not coming in, but potassium is, causing even a greater polarization from one side of the membrane to the next. Here's another question. Adding a poison that specifically disables the sodium-potassium pumps to a culture of neurons will cause, what would happen if we didn't have the sodium-potassium pump doing its job? The resting potential would drop to to 0 millivolts. The inside of the neuron would cause the inside of the neuron to become more negative relative to the outside. The inside of the neuron to become more positively charged relative to the outside. Or the sodium to diffuse out of the cell and potassium to diffuse into the cell. And the answer is A. If we don't have sodium-potassium pumps returning, we're going to get a membrane potential. The resting potential would go to a 0. Let's look at some adaptations. What makes the membrane of a neuron able to specialize in conduction? The speed of the action potential increases with the axon's diameter. The second, in invertebrates, the axons are insulated with myelin sheaths. That allows us to be faster. Those are secreted by glial cells or oligodendrites in the central nervous system and Schwann cells in the peripheral nervous system. So those adaptations allow for an increase in speed. Here you can see an axon that is surrounded with layers of myelin, which is a protein. So these Schwann cells have to be really good at protein synthesis because their job is to secrete myelin, which acts as an insulator. If we cut this axon across, then this image down here is a cross section, so you can see the layered effect of the myelin surrounding this axon. And that way the only place where we're going to have the depolarization event occurred would be in the nodes that exist between these bundles of sheaths there and in between the Schwann cells, so that would allow conduction to happen very rapidly because it only has to occur, depolarization only has to occur in the nodes, which would allow for an increased speed and transmission. And so we see this sort of jumping along or saltatory conduction, and that saltatory conduction is an advantage to cells that are trying to have a really fast response. And so the presence of a myelin sheath is actually going to even make the conduction of the action potential occur even faster. Next time we're going to look at what happens once the impulse gets all the way down to the end of the axon. And so we'll see how the actual transmission to the next cell occurs. So see you next time.